How to prepare animal specimens using paper cards. The paper cards are intended for collection, storage, and transportation of animal specimens for molecular biology applications. The kit has 25 cards placed in separate envelopes. One card is for 4 biological specimens. The sample is applied onto the corresponding card circle, framed by the dotted line. The sample should cover the whole inner perforated part of the circle. The cards are intended for use with whole blood samples, organ and tissue samples, washings and swabs. How to collect, store, and transport animal whole blood. Thoroughly mix the blood sample. Do not use heparinized blood samples. Pipette 50 microliters of the sample onto the corresponding card circle using a new tip with filter for each sample. How to collect, store, and transport animal organ samples. Separate a tissue fragment using scissors and tweezers to make a visible impression. To do this, tightly press the tissue fragment to the corresponding circle on the provided card. To prevent contamination, sterilize the tool edges with the flame of an alcohol burner after each sample and clean the tools with disinfecting wipes. How to collect, store, and transport washings and swabs from animal mucosa. Thoroughly mix a sample. Pipette 50 microliters of the sample onto the corresponding card circle using a new tip with filter for each sample. If the sample spreads outside the dotted line circle, repeat the application procedure using another circle. After applying the sample, leave the cards to air dry at room temperature. Avoid direct sunlight, wetting, or extra heating of the cards. After drying, label the card appropriately. Put the card with the applied samples in the envelope and label the envelope. The cards are now ready for long-term storage, transportation, and extraction of nucleic acids. Use the inner perforated part of the circle for the extraction of nucleic acids. Press out the perforated part of the circle into a labeled tube using a disposable tip. Push the paper to the bottom of the tube. The samples are ready for DNA and RNA extraction. We recommend the following kits manufactured by Vector Best for the extraction of nucleic acids. For manual extraction, Real Best Vet Gamma. For automated extraction, Real Best Vet Beta in combination with an activating solution. How to pre-treat a sample using the inactivating solution for nucleic acid extraction with the Real Best Vet Beta Kit. Before work, heat the vial with the inactivating solution for 30 minutes at 50 to 60 degrees Celsius to dissolve the precipitate. Mix the solution and keep it at room temperature for at least 10 minutes. Using a new tip with filter, for each sample, add 500 microliters of the inactivating solution to each tube without touching the walls. Vortex the tubes. Incubate the samples in a thermal shaker at 1,300 revolutions per minute and 65 degrees Celsius for 20 minutes. After the incubation, centrifuge the samples at room temperature and 13,000 revolutions per minute for 1 minute. Use 100 microliters of the supernatant for the extraction of DNA or RNA using the Real Best Vet Beta Kit. Inactivated samples can be stored or transported for future PCR analysis.